Hello. We're going to talk about how DNA replicates. Now, we did talk about it a little bit before when we talked about the cell cycle because DNA replication actually occurs during the cell cycle. So now you're actually going to learn about how it really happens. So it occurs in the nucleus of a cell because that's where the DNA is. It happens during the S phase of the cell cycle. So remember, during interphase, you had G1 where the cell grew, and then the S phase where the DNA duplicated, and then G2 before it went into mitosis. So the S phase, or when the DNA duplicated, is what we're talking about here. And the reason that it happens is so that when the cell divides, each new cell is going to get an exact copy of its DNA. So that's why the DNA has to replicate. So replicate means basically to duplicate, to make a new copy that's exactly the same. Now, DNA replicates through what's called a template mechanism. Now, if you remember, DNA is shaped like a double helix. It looks like a ladder twisted around itself. So what it actually does is it splits apart those two strands and uses the two old strands to make the two new strands. So one of the strands acts kind of like a negative for building the other one. And it's really simple, actually, because there's base pairing rules. So adenine's going to pair with thymine, and cytosine's going to pair with guanine. So whatever's on this old strand, it's going to pair the opposite thing on the new strand in order to make the new DNA molecule. So DNA replication is the process of copying a DNA molecule. And like I just said, it's really simple because of that double helix structure. Now, when DNA is replicating, the first thing you have to do is you have to split the DNA apart. And where that splits apart is called a replication fork because it's kind of like a fork in the road. It's where it splits into two. So a replication fork forms and the strands split apart. And that's done by an enzyme called DNA helicase. If you look at it, helicase almost looks like it has the word helix in it. It unwinds the double helix. And then another enzyme called DNA polymerase puts new base pairs on to make the new strand. Now, remember from way back in the fall when we were talking about monomers and polymers, a polymer is a long string of monomers. Well, DNA polymerase puts the monomers on to make a long chain. So those are two enzymes that you need to know. They're going to be quiz questions, so you're going to want to write both of those down. DNA helicase unwinds it. DNA polymerase puts new nucleotides on. Now, as this is happening, it's not just happening at one end and going all the way to the other end. It actually starts at different spots in the DNA at the same time and makes things that look like little bubbles. So it begins at spots called origins of replication. Origin just means beginning. And they're sites that it'll start at the same place every time. So it's actually going to copy in both directions at the same time, making those bubbles. And it's also going to be copied on both strands at the same time. So it can happen very quickly because it's starting at so many different spots at the same time and going in both directions. So what you're seeing here is actual DNA being replicated inside a cell. So this blue fuzzy thing here is DNA helicase. It's actually taking the double-stranded DNA and unwinding it. And you see two strands coming off of it, one to the bottom and one off to the side here. And they're actually new strands being formed from the old strands. Now, this one in the middle here almost looks like you're crocheting because it's, doing some and then pulling in and doing some more. That's because it has to be replicated backwards. So it's pulling some and replicating and pulling some and replicating. But your original each one then is going to have one new strand and one old strand. And the green part here is the DNA polymerase. That's the thing that is adding the nucleotides onto the DNA. So now when the DNA is replicating, remember that it uses its base pairing rules in order to create the new strand. So what we're going to do here is we're going to use the base pairing rules to create a complementary strand for what you see here. So the strand we have is AA, CG, TG, CA. So remember, adenine pairs with thymine, cytosine pairs with guanine. So adenine pairs with thymine, so under this A you would write a T. Another adenine is going to get another thymine. Cytosine pairs with guanine, so C would go with G. Guanine pairs with cytosine, that would be a C. T pairs with A, that's thymine pairing with adenine. G pairs with C. C pairs with G. And A pairs with T. So all you really need to do when you're given a sequence is match it to its buddy. So go ahead, write a podcast question that you still have a question about, and I'll see you in class.